So we're going to be covering the Agilent 1260 mols, and more specifically, we're going to be looking at the fundamentals of light scattering and case studies for macromolecules using our new 1260 mols. As a reminder, my name is Steve Rosa, and I'm one of the GPC SEC product specialists here at Agilent Technologies. What I'm going to do next is just run through a brief table of contents before diving into the content. So just looking at what we're having to look at today is we're going to start off with the history of GPC and SEC, sort of dive into where this technique came from, where it was initially used, and where we have some limitations, and then use that as a springboard to jump into light scattering. We'll talk about the fundamentals behind light scattering, why it works and how it works. And from there, we'll link everything together with just applications that we see our customers using light scattering for, just some interesting points and maybe some non-traditional approaches to using light scattering, but things that are interesting nonetheless. And we'll finish off today with a question and answer session with Lorena and myself. So when we talk about GPC and SEC from a traditional standpoint, what we really need to start is in the materials world, and specifically looking at polymers. For those who are unfamiliar, polymers are macromolecules that are composed of one or more repeating subunits. These subunits are called monomers. Through polymerization of these monomeric units, you end up with the polymer backbone. Polymers exhibit very different physical properties compared to monomers, and those properties of the polymers themselves depend on the length of the polymer chains within the polymer as well. The presence of small amounts of very long or very short chains can have a considerable effect on the bulk properties of your final material, and finding a way to characterize these is where GPC and SEC initially took off. So to dive a bit deeper into what I was saying, what we see here is if we just look at I'd say the most basic polymerization that we can take a look at here, taking the monomer of styrene and creating polystyrene from it. Now, when you do a polymerization, you're targeting a specific molecular weight of your polymer, but in reality, what you end up with is a statistical distribution, much like we see on the left. In this case, with this polystyrene, if we also look down on the right, you'll see that using mass spec, we can see that there are individual peaks for each of these different MERs. So while the target was likely around 1400 Dalton, what you can see is that we have a wide distribution ranging from about 893 Dalton all the way up to almost 2600 Dalton. We have the statistical distribution. We need a way to describe what different properties on these materials are. And this is where we see things like the number average molar mass and the weight average molar mass. So what we're going to do is walk through a couple example calculations of both this MN and this MW using these two pile of marbles up above. So just for reference, the pile on the left has 500 1-gram marbles. Those are shaded in blue. And there's two marbles that weigh 250 grams. Which gives us a total weight of 1,000 grams worth of marbles. On the right side, what you'll see is we have 400 marbles that weigh 1 gram, and we have 100 marbles that weigh 6 grams, giving us an equivalent weight to that first pile of 1,000 grams. And what we're going to do in the next couple of slides is really illustrate how a few large outliers can influence the different ways that these calculations come out. The first of these properties we're going to take a look at is the number average molar mass, or MN. MN is the sum of all of the mass fractions multiplied by the number of molecules in these fractions divided by the sum of all of these molecules across the entire fraction range as well. The important takeaway from this slide is if we look at the pile on the left with the two really large marbles in there, we can see that having these in here leaves us with an MN of 1.99. If we look to the right with the more evenly distributed marble pile, we can see that the MN is 2. Having these two really large outliers doesn't have that large of an effect on the MN number. While what we'll show in the next slide when looking at the next thing that we're calculating, which would be the MW, or the weight average molar mass, is that these outliers are going to have a much more significant impact on the numbers that are calculated. Now what we're going to do is take a look at the weight average molar mass, or the MW. As you can see, if we just look across the values here for these two marble piles, you'll note that when we compared to the MN from the previous values, we see that MW on the pile to the left is 125.5. When we're looking at the number average molar mass, it was 1.99. Looking at the pile on the right, we'll see that the MW is 4. While on that previous slide, we had an MN of 2. And the big takeaway here is that having just a few large outliers and a much less narrowly distributed sort of sample will lead to a gigantic influence in raising the MW of the sample that's being looked at. And from here, what we can do is find a way to describe how these are distributed, which we'll talk about more in the next slide. So after we've determined an MW and an MN, what we can do is put them in a ratio over one another, which we refer to as the polydispersity index. Polydispersity index, or PDI, is a measure of heterogeneity in the piles of marbles here. So we can see that looking at the left, where we have this large MW number due to these two large outliers, we end up with a dispersity index of 63. Looking at the right, where we had a MW of 4 and an MN of 2, we can see that the dispersity index is 2. And what that is essentially telling us is that the pile on the right is much more homogeneous in composition than the pile on the left. Now, PDIs change depending on the different types of polymerization techniques that you're using. And down below on the right, you see a typical range of PDIs we would expect across a whole different range of polymerization techniques. For free radical polymerization, we would expect to see PDI somewhere between 1.5 and 2. For a controlled or a living polymerization, we would expect a much narrower PDI, so somewhere around 1.01 to 1.3. A step growth polymerization will get us a PDI right around 2, and then polymerizations with several active species can be, much greater than 2, and in some cases up to, 8 and 10 in terms of dispersity index. Now that we know what these different parameters of MN, MW, and PDI are, the question is, well, how would we get these? Where does separation come from? And when we're talking about separation in GPC and SEC, the driving force of separation here is a concentration gradient between the mobile phase that's inside and outside of the pores within the column. Now, here's just a view on the right-hand side of an SEM image of one of these pores, but the principle works like this. The sample is prepared as a dilute solution and eluents injected into the system. The injection of this material enters the column, which is packed full of these different particle sizes and porosities. Large molecules aren't able to permeate all of the pores and have a shorter residence time in the column, and thus elute first. Small molecules are able to permeate and penetrate into these pores of the column, and therefore take longer time to come out. And they come out after these larger molecules. In this case, what we get here is macromolecules that are separated according to their hydrodynamic volume. So the largest will elute first, and the smallest will elute last. What I'll show here is just this separation principle sort of as it happens. So what we have here is isocratic flow and the stationary phase with all these different pores. And what you'll see is that large molecules will be the first thing that come out. Small molecules will be the last thing that come out. What this is an entropy-driven process. Now, what we can do is contrast this against something like an HPLC type separation, where here we have chemistry on the stationary phase that we're trying to interact with. So what that allows us to do is do something like, in this case, a reverse phase type separation, where we'll have polar things come out first and nonpolar things coming out second, or last in this case. But what we can do is also change up our separation by changing column chemistry and column conditions. Now we can also separate in what we call the normal phase, in which we would have the exact opposite happen, where nonpolar molecules are going to be the first off of the column, and things that are more polar are going to be eluting later. And what this gives us is what we would call an enthalpy-driven interaction. And this sort of really highlights the difference in mechanisms between SEC and the other HPLC type separations. Given that SEC is entropic, you're really looking to have no interactions with the column itself. On top of that, there really isn't much you can do in order to increase separation, aside from adding column length to therefore increase separation efficiency, since the pore volume available is directly responsible for how well you're going to get separation. Now, we've briefly touched on the concept of hydrodynamic volume, and this slide really highlights what we're getting at here. So macromolecules in solution behave in various different ways. In the most simple case that we can look at on the left-hand side, what we're looking at here is actually polystyrene, but in different mobile phases. So you can see that in methanol, there's quite a poor solvent for polystyrene. The hydrodynamic volume appears quite small. But as we start moving towards THF and chloroform, that hydrodynamic volume of polystyrene is going to increase quite substantially. The other thing that's important to note is that different polymer types in the same solvent are going to have vastly different swelling properties as well. And this is where you can really trip yourself up when doing GPC and SEC by conventional calibration. So in this case, say we calibrated off a polystyrene standard, and that was what our curve was built off of. In this case, the polystyrene at this hydrodynamic volume is showing a weight of about 20 kilodalton. Now, if we injected a polyisoprene that was 15 kilodalton, or a PMMA that was 24 kilodalton into this column, based off of these hydrodynamic volumes, we would get a mass estimate of 20,000 kilodalton, given that these hydrodynamic volumes are equivalent to that polystyrene. Having seen that last example where we can have different molecules with different chemistries occupying the same space and therefore giving the same mass results using conventional GPC or SEC, we might find ourselves asking, well, how could we get the actual molecular weights of these materials if that's something that's important to the research or the problem we're trying to solve? And the answer we're going to look at here is going to be with light scattering. So we'll dive into the physics behind this and how we're actually able to distinguish these individual or absolute molecular weights separate from their hydrodynamic volume, which is the principle that governs separation. When we talk light scattering in macromolecules, what we're talking about mostly in this presentation is actually going to be static light scattering. In this case, we're going to just describe how static light scattering works. So with static light scattering, we have a coherent incident radiation source. It's usually a laser. And what this does is when shined into a flow cell, interacts with components of the backbone of the material in a small scattering volume. The excitation of this material results in scattering and re-emission of the radiation at the same wavelength and frequency, but at a differing intensity. Measurement of this differentiation in intensity of the scattered radiation against the incident source is what allows us to make the calculation of the molecular weights, and if the molecule is large enough, a few other parameters as well. In theory, what we were talking about on that last slide ends up looking like this. This is the light scattering equation itself. You can see there are a whole numerous bunch of components that go into this. We have things like K star, which is the optical constant, as well as this particle scattering factor, which is what we have in here when molecules get really, really large. I'm just showing this to highlight that light scattering really is a complicated thing, and there are a whole bunch of different parameters that we need to consider when we are talking about getting accurate light scattering. And luckily, though, we're able to simplify this in some cases. So we'll look at one of those examples coming right up. So luckily, in most cases, especially when we're talking about isotropic scattering, we're able to simplify this equation. In the case of isotropic scattering, where the molecule looks the same no matter what angle you're viewing it from, that particle scattering factor becomes equal to 1 and therefore can be removed from the equation. What we're able to do then is simplify to what you see below. What you'll see here is that we have the optical constant still, as well as the need to know the concentration. We also have the scattering intensity, which is simply the measure of light emitted versus light returned. And then you'll see that there's molecular weight, which is the result that we're interested in. You'll note DNDC is also in here, so you need to know the sample's DNDC and its concentration if you're looking to do best practices. But with the WinGPC software, knowing one of them is sufficient. That simplified equation for isotropic scattering is nice, but we're not always going to have isotropic scattering in what we're looking at. When we start looking at molecules that are large in solution and look different from different angles that they're viewed at, also known as anisotropic scattering, simplification of the light scattering equation isn't possible, and we need to have this particle scattering factor thrown back in the mix. The important thing to note within the particle scattering factor is this root mean square, or as most people refer to it, the radius of gyration, which is a measure of size of the molecule in solution across the available angles. This radius of gyration isn't a measurable number unless you have anisotropic scattering. So if you have an isotropic scatterer, and typically we say these are usually located right around under 10 nanometers, you're not going to be able to get a radius of gyration calculation from using multi-angle light scattering. Now, as these molecules get larger in solution, and we start leaving that 10 nanometer space behind and move towards 20, 30, 50, 100 nanometers, then radius of gyration is something that we'll be able to calculate with more and more precision. Here's just a visual example of what these last two slides really are talking about. So if we look at that top example, where we have the particle size of less than 1/20th of the wavelength of the laser of the light scattering detector, you'll see an example of isotropic scattering. So you'll see that it doesn't matter really where you look at this material from. It's going to look the same from all given angles. The important thing to note with the anisotropic scattering regime is now as we start moving down to examples 2 and 3, as these molecules get larger and larger, we see more and more angular dependence within these molecules themselves. This allows that particle scattering factor to be thrown back into the equation and allows for us to calculate a radius of gyration of that material. So what does light scattering give us? Well, with light scattering, since we're directly measuring molecular weight, we can get the absolute molecular weight independent of the hydrodynamic volume that we would see with the weights that we get from a conventional GPC. Light scattering can get us information on branching by looking at, say the radius of gyration. If we have a molecule where we have a decrease in the radius of gyration while molecular weight stays the same when compared to another molecule of the same chemistry, that's something that typically is indicative of a branched material. We can also get polymer conformation information from this. So as you can see here in these examples, these molecules are more compact because of branching, but not necessarily lower in molecular weight. And if we look on the right-hand side, looking at these polyethylene examples, you'll see that given the same molecular weight, that radius of gyration is actually smaller for these branched samples. So looking at this, the blue sample would be more branched than the black sample, would be more branched than that linear PE that we know up at top. So knowing all this, then the next question would be, well, when are these advanced detectors needed? Well, GPC and SEC with a relative calibration can provide information in terms of the distribution of chain lengths and the relative molecular weights. And sometimes that's just enough. You don't really need to know more of these advanced parameters. But if you're interested in knowing the molecular weight, be it accurate or absolute, if you're looking for information on the macromolecule's size in solution, or if you're looking to extrapolate information on the macromolecule's shape in solution, this is when you need to start thinking about getting light scattering, or in some cases viscometry, into the mix. As these are going to allow you to dive much deeper into looking at these material characteristics. Having covered the history and why GPC and SEC were developed, and talking about sort of the fundamentals in the physics and what light scattering can give us, it's time now that we're going to delve into some applications, looking at what customers and what we're doing with light scattering, and highlight where it can be used and the potential it has within a lab. So the first, and I would say the most obvious use for light scattering would be in terms of looking at absolute molecular weight and quantifying aggregation. And what we'll do now is show a couple examples of where we see this in practice. So in this slide here, what we have is essentially let's say a more complete version of this macromolecules in solution slide from earlier. What you'll note is here we now have three different materials. We have an ovalbumin, we have polyolane, and we have polyethylene oxide. All of these are right around 45 kilodalton. Granted, they're all very different size in terms of their repeat units. We're looking at 385 amino acids for ovalbumin. We're looking at a 275 unit repeat for polyolane, and we're looking at about 1,000 repeat units for polyethylene oxide. Now, looking over on that right-hand side at these different chromatograms of these, you'll see that they all come out in very different spots in the column. And this is due to the fact that they all have very different hydrodynamic volumes in solution. That PEG, being 1,000 units long, is going to be a much longer molecule and occupy a larger hydrodynamic volume than something like that ovalbumin, which is going to be relatively compact in solution like most proteins are. Now, if we were looking at these three materials using a conventional calibration, we would end up with three different molecular weights. So if we had calibrated off of, say, polyethylene oxide, we would get 45 kilodalton for the PEO, but the polyolane and the ovalbumin's molecular weights would be vastly underestimated due to the fact that, we're just looking at hydrodynamic volume here. Now, by getting light scattering involved, what we're able to actually do is get the exact values that we're expecting to get off of these. So we can see that the PEO is 45 kilodalton, the polyolane is 45 kilodalton, and the ovalbumin is 44.3 kilodalton, despite the fact that they have different hydrodynamic volumes. We're looking directly here at molecular weight. On top of being useful for just determining absolute molecular weights, a nice thing that light scattering gives us the ability to do is to look at these low concentration, high molecular weight aggregates, or even just mass fractions within samples. And this really just has to do with how the detector responds to these different materials. So if we look at just on the left here, what you'll see is in green is a concentration detector. In this case, it's a refractive index detector. And what you'll see is that it's responding purely to concentration. So looking at that, we can determine that whatever is the highest molecular weight of these four materials is at about half the concentration of the three that follow it. Now, looking above at the light scattering detector, what we see is that the signal intensity is directly proportional to molecular weight here. So that material that's half the concentration is of a high enough molecular weight that it's really giving a good light scattering signal. And as we start decreasing in molecular weight, we see signal quality start to degrade until eventually, with that really small peak hovering around that 10-minute elution volume mark, it's actually so small we really don't see anything within that light scattering detector. But given this allows us to leverage moles to look at, again, these low concentration, high molecular weight fractions or aggregates within materials, which I'll show a couple examples of coming up. Now, looking at this, what we have here is a gelatin. You can see that if we compare the response from the refractive index to the light scattering detector, what we see is a vastly different profile. While we see this little shoulder in that refractive index detector up in the high molecular weight regime, somewhere starting around 10.5 minutes. What we see looking at the light scattering detector is this massive response to these higher molecular weight portions of this. In this case with gelatin, it's nice to see that maybe we do have high molecular weight stuff up in this sample that we would have otherwise missed. But where this really becomes important is when we start thinking about looking at biologics in solution. So what we have here is an oligonucleotide that we were looking at. In this case, it's just an mRNA sample. But what we can see is if we look at the concentration detectors, so blue being UV at 260 and red being RI, is that as we start to get to the dimer and tetramer and higher order aggregates, which are in much lower concentration when compared to the monomeric peak we see at about 7 minutes, is we see a decrease in the signal intensity for these concentration detectors. Now, looking at that light green trace, which is the 90-degree angle off the light scattering detector, what you see is we have a much higher response to these, even though they're in low concentration, and that's due to the fact that they have this really high molecular weight, again, that the light scattering detector is quite sensitive to. Continuing on with this theme of looking at different aggregates, in this case, what we're looking at here is a MabThera mab. So if we look on the left, what we have is a completely unstressed MabThera. We'll see that it has a molecular weight of 144 kilodalton, right around what we would expect. But on the right-hand side now, we have this MabThera, but we've stressed it. And what you'll note is that, again, comparing the concentration signal in blue, which is the UV at 280 nanometers, and the light scattering detector at 90 degrees, is that we have a much higher response to these high order aggregates from the light scattering detector than we do the concentration detector. And this allows us to screen for really low concentration, high molecular weight aggregates in biological samples. And again, just backing it up with the math here down below, this is the case because we're looking at molecular weight directly. So as we get higher molecular weights, we would expect higher signal intensity given the same concentration. In this case, we have low concentration, which is where the UV falls short, but the moles picks up what the UV missed otherwise. And so with this, I'd like to talk about one of the collaborators that we have at Virginia Commonwealth University. We're working with Professor Fabio Gomes, and he's using the 1260 moles in conjunction with mass spec to investigate adeno-associated viruses and their aggregates. Now, sort of going through from back at the very beginning with that video that we started with, his detectors are a diode array detector. He has the 1260 moles, and he has a bioinert fraction collector. And so far, what Professor Gomes has been doing is he's using the moles to see the low concentration, high molecular weight aggregates that are within these AAV samples. From here, he sets his fraction collector to collect that region of his chromatogram. From there, he takes these fractions and he injects them straight into mass spec. It should be noted that he's running a mass spec friendly buffer on everything to be doing this. So he's running all of his AAV separations in SEC with an ammonium formate salt. Just an example of what that might look like using this AAV2 data that we generated at our lab in Lexington. You can see that up around that 9.5 to 10.5 minute mark, there's this high molecular weight peak. It's especially obvious when looking at the light scattering 90-degree angle, which is the blue trace within this chromatogram. So he's setting his fraction collector limits right around that time frame, and from there, going to mass spec. So the second thing that that light scattering is quite useful for is determining the size and solution of a molecule, and we'll kind of dig into a few examples of this as well, like we did with molecular weight and aggregation. As a reminder of these two terms from earlier, isotropic and anisotropic scattering, what we have here are the two different regimes that we expect scattering to be in. So in isotropic scattering, the intensity of scattered light is independent of the angle it's measured from. It's going to look the same, whether you look at it with 2 angles, 3 angles, 20 angles, 100 angles. It's all going to scatter identically. It's too small to have angular dependence in solution. Anisotropic scattering, as you can see, is different. The scattering centers lead to interference. This different scattering intensity is measured at different angles, and this is what allows us to calculate a radius of gyration in solution. And the reason I'm bringing this up, again, is just to highlight what you need to have in your sample in order to calculate size using static light scattering. And why is that? Again, just to cover from earlier in the presentation, the isotropic version of the light scattering equation is nice and simple. We have just a few terms here, but when we start talking about anisotropic scattering, we introduce this particle scattering factor, and you'll note within there is where radius of gyration is sort of living. So without anisotropic scattering, without angular dependence within our samples, we have no way of measuring radius of gyration using static light scattering. Again, just reminding you of some of what these other constants are as well, but the main point here being, if you're looking to get size and solution information from static light scattering, you need to have angular dependence within your material. Otherwise, you'd have to consider going to some other sort of characterization technique to get an RG below, say, 10 nanometers, like small angle X-ray scattering. We've been talking exclusively about static light scattering so far throughout this presentation, but another form of light scattering does exist, which is dynamic light scattering. So with static light scattering, what we're doing is we're measuring the time average intensity of scattered light at one or more angles as a function of the angle theta. What this gives us is the molar mass and structure of molecules. So we get MW. If we have anisotropic scattering, we get the RG, and we can get the virial coefficients. Those virial coefficients just describe molecular interactions, be it with itself or with the solvent. Dynamic light scattering is another form of light scattering that some people do, and this measures the time-dependent intensity of scattered light. And what this allows you to do is calculate the radius of hydration. It's important to note here that when we look at RH, we don't have that same limiting factor of needing anisotropic scattering to calculate hydrodynamic radius. But the final thing we'll look at here is actually using this radius of gyration and this hydrodynamic radius and combining these two different values to extrapolate the shape of a material in solution. So what do these radii mean? So we know that from static light scattering, we can determine the radius of gyration, and we know from dynamic light scattering, we can get the radius of hydration. Both of these terms are different. Looking at this lysozyme molecule below, we can see that drawing out these different radii, hydrodynamic radius and radius of gyration occupy different areas in space. And this is because in real life, these molecules don't conform to a static hard sphere model. But what this allows us to do is take a ratio of the radius of gyration over the over the radius of hydration, and get what we call the shape factor. And this shape factor can indicate what that material is behaving like in solution. So if we look at something and we see a value of zero point 0.7, we know that it's acting quite like that hard sphere model. As we start moving up towards 1, 1.2, 1.4, what we see is a material going from a sphere to more of a coil to becoming much more rod-like in solution. So looking at an example of this, we're just going to take a look here at thyroglobulin. So if we take a look at a monomer of thyroglobulin, we end up with a radius of gyration of 12 nanometers, we get a radius of hydration of 9 nanometers, and we get a shape factor of 1.33. So we're not quite rod-like, but we're certainly in a transition state between an ideal coil and a rod-like structure. Now, interestingly, if we get a dimer of thyroglobulin, what we see here is that we have a radius of gyration of 16 nanometers, a radius of hydration of 14 nanometers, and a shape factor of 1.14. So what this tells us is these two rod-like structures aggregate together and form almost a coil-like dimer unit. To look at another one of our collaborators, in this case highlighting architecture, currently we're working with Professor Todd Emmerich's group at the University of Massachusetts Amherst, and they're using mols to study complex polymer architecture. Specifically, they're looking at bottle brush polymers. Due to their irregular shape, accurate molecular weight info isn't possible using conventional GPC and SEC type calibrations, but also as these materials grow, they behave differently. So you'll see this transition from something that behaves more like ideal coil that becomes rod-like and then shrinks back almost towards that ideal coil again. So by using shape factor and by getting the absolute molecular weight, they're really maximizing what they can do with multi-angle light scattering. In their case, what they're working with is a refractive index detector. They're working with a UV detector in the 1260 mols. Their RH calculations are coming off of a benchtop DLS, and we'll show some published work from here. Taking a look at what the Emmerich group is doing with these bottle brush polymers, we'll take a peek down here at figure C. We can see that initially, when we have 19 of these bottle brush repeat units, we have a shape factor of 1.18. This is indicative of something that's behaving like an ideal coil in solution. As we start increasing, as you'll see on the bottom on that x-axis there of NBB, to 47 of these bottle brush units, we see, again, a shape factor of 1.20, still in that ideal coil sort of space. It's when we take a look at this third point here, when we have these 92 repeat units, and what we see now is that we have a shape factor of 1.51, which is indicating that this material is beginning to act really quite like a rigid rod. And interestingly, as we increase these repeat units even further towards 190, what we'll see is we have a decrease in the shape factor here. So from 1.51, we drop to 1.40. While still in that rigid rod regime, we can see that as this polymer is growing larger, there are more degrees of freedom that are coming across this backbone and allowing some extra flexibility in there, which is likely leading to that decrease in the overall number associated with that shape factor.